Hello, this is Dr. Lawson in the second in the series of How to Better Study for Dr. Lawson's lectures. This is the lecture on the control of the human cell cycle, which we had earlier today. The control of the human cell cycle lecture covers material that is poorly covered in your textbook, so you will need to use things like Wikipedia and Google searches for reading material on this, as your textbooks have an insufficient background in this area. However, I would recommend the Wikipedia article on cell cycle control. It is very complete, although it is extremely detailed. I would like you to remember the objectives primarily focus on the events and the control of the events that mark each of the checkpoints of the cell cycle. Just knowing the cell cycle is insufficient, as is just knowing all of the names of all the proteins is insufficient for this lecture. You need to know how to control each of these proteins. This slide was a reminder of the cell cycle, and this slide told you how we activate cyclin-dependent kinases and who their cyclin partners are. Make sure you know both the CDK and its cyclin. Know that the CDK, when it comes with our cyclin term that comes with a cell cycle phase attached to it, like G1 CDK or G1 cyclin, that means the complex of a CDK cyclin. And the G1, for example, would be cyclin D with either CDK4 or CDK6. You do need to know each of these CDK complexes, knowing which cyclin and which CDKs go together and which phase of the cell cycle they control. You need to understand that the cyclin CDK complexes are very strongly regulated themselves in both their production and their destruction on a cell cycle basis. The G1S cyclin, which is cyclin E, is produced in mid to late G1 and diminishes immediately after the G1 checkpoint is passed. The S cyclin begins to grow in late G1 and persists throughout the cell cycle until the destruction at the metaphase-anaphase checkpoint. Likewise, the M cyclin is only turned on in G2 and is destroyed at the M phase, metaphase-anaphase checkpoint. Make sure you are very well aware that DNA doubles in the cell cycle. This graph is an important graph. It does show up on exam questions from time to time. You should be able to align the cell cycle phase with how much DNA is in the cell at any given time. Cyclins are, you need to know that they have 4 levels of control. They are gene expression, phosphorylation, and inhibition by the cyclin-dependent kinases. You need to also know that they can be ubiquitinated and destroyed after ubiquitination. This is the way all CDK cyclin complexes are degraded when they are taken back apart. This next 2 slides remind us of those pathways. There's the phosphorylation. Hyperphosphorylation inhibits the CDK, whereas the original activating phosphorylation activated. Removing inhibitory phosphates can also reactivate it. This is the method that is used predominantly in G2 to regulate the checkpoint. CKIs are used by direct suicide inhibition using the CKI protein. This is the method used in G1. And in the M phase checkpoint, we use the complete ubiquitination of the CDK cyclin complexes to facilitate their destruction. We always go into G1 first and then into G0 We can leave G0, we will always go back into G1. Make sure you understand that in early G1, the cyclin that's important is cyclin D. Cyclin D is driven by mitogens, and mitogens like MYC activate certain proteins that then result in the activation of a very important protein called P53. P53 in its normal state, when it's activated, propels the cell into the G0 state. It also can elicit the cell to go into cell cycle arrest or apoptosis, depending upon the kinds of signals that have sent the cell into G0. Mutate either MYC or P53, that this leads to a cancer state in many cases. In fact, most cancers have both MYC and P53 mutations. They also typically overproduce those mutated forms of those two proteins. Those mutations usually activate MYC or they inactivate P53. The MYC in the when P53 is inactivated actually helps to further propel the cell into cell cycle. You need to make sure that you understand there's a number of factors that regulate both cell growth and cell division. They do it through the same sort of signaling cascades that Dr. Peterson talked to you about earlier this week. The one that is most important for this lecture is receptor tyrosine kinases acting through the MAP kinase cascade, producing MYC, FOS, and Jun. Overexpression of these growth factor receptors, particularly ones that are also mitogenic, can result in cancers as well, and that is seen in lung adenocarcinomas. The way these receptor tyrosine kinases work is they work through MAP kinases. The MAP kinases second messenger system activates genes that in turn activate the production of MYC. MYC then activates the production of additional genes. Those genes involve the activation of cyclins, and the cyclins then bind their CDK partners. They can then act on the cell. The most important one in G1 is the G1 CDK cyclin. That would be cyclin E. Cyclin E can then phosphorylate proteins like the RB protein, which is a binding partner to that very important transcription factor we've already learned about called E2F. When RB protein is phosphorylated, it can no longer hold on to E2F. E2F, you will recall, is a transcription factor. That transcription factor then causes the production of more cyclins. As more cyclins are produced, we get more RB protein phosphorylated and inactivated. This is a positive feedback loop, pushing the cell further and further toward DNA synthesis. A disease state that is associated with this RB pathway is retinoblastoma. It's a type of eye cancer. When retinoblastoma gene or RB gene is mutated and overexpressed, it releases this control and there's no repression. RB's normal job in the cell, remember, is to act with E2F to inhibit the production of DNA synthesis requiring genes. So when RB protein can never bind E2F, we can never stop DNA synthesis from occurring. You need to know this slide in its animated form. The animation is very critical. We have second messenger systems that act on the cell by binding their hormones. They act through their second messenger systems, creating transcription factors. Those transcription factors result in the increased production of cyclins, initially cyclin E, but eventually cyclin A. Those cyclins are translated in the cytosol, folded, and brought back into the nucleus where they associate with their CDK binding partner. They are then activated using the CDK activating kinase. That activated kinase can now act on RBE2F complexes releasing more E2F by phosphorylating RB, allowing the E2F to now go and act on more genes. As it acts on more genes, it will turn on all of the genes required for S phase. As this progresses, this is a positive feedback loop, obviously. This continues until we are ready to go to the next step and start replication. There is, however, a brake system for this pathway, and that is if the DNA is damaged, we have a system to turn off this cyclin activation of E2F. And the way we do that is use a cyclin dependent kinase inhibitor like P27 or P21. They bind the CDK cyclin complex. When they bind the complex, they then cause it to go to the cytosol and become ubiquinated and destroyed. This positive feedback loop is in direct competition with this inhibition loop, and as soon as the DNA damage is resolved, the positive feedback loop wins and we go on into DNA replication. This is a reminder of how DNA replication begins. This is a slide reminding you how the cell senses DNA damage. The DNA damage causes the activation by phosphorylation, a different pathway than the MYC activation of P53. P53 that has been phosphorylated is a transcriptional activator of a gene called P21. P21 is a CKI and is used to inhibit both the G1 and the S-CDK cyclin complexes. The way it inhibits them is by suicide inhibition, and then it also causes the ubiquinization of both the inhibitor and of the cyclin. S phase also has a setup so that the sister chromatids are tied to each other. You need to know that. G2 checkpoint is considerably different in that the pathway here is regulated in a different way. The regulation after E2F is making mitosis-specific genes, and the inhibition here is by phosphorylation in response to DNA replication. If DNA replication is incomplete, Wee kinase or Wee1 hyperphosphorylates the CDK until the DNA replication is complete. As soon as DNA replication is complete, CDC25 can reactivate the CDK cyclin complexes and allow us to finish making all the proteins required to go into M phase. Both S-cyclin and M-cyclin can be inhibited in this pathway. Finally, in the M phase checkpoint, we have this protein called APCC. APCC is phosphorylated by CDK cyclin complexes and activated. When it is further activated by another protein, it can now bind proteins that are important for metaphase. After the DNA has lined up on the metaphase plate, APCC can bind this complex of securin and separase. It binds the securin, holding it, causing it to release separase. Separase is released. Separase can then go and cut all of the cohesin molecules that are holding the sister chromatids together and allow the sister chromatids to come apart in anaphase. As the APCC binds securin or it binds the CDK cyclins, it can actually then cause the ubiquinization of both of those. APCC is responsible for the final destruction of all remaining cyclins in the cell at anaphase, and that this can be inhibited by the failure of the spindle to attach properly or the failure for the metaphase chromosomes to line up properly. At that point, you inhibit the activation of APCC. If APCC isn't activated, you cannot separate the sister chromatids. Lastly, remember what causes the cyclins to be activated. Remember what causes them to be inhibited. What are the signals from the cell, from outside the cell, and how do they interact with each other. Remember that favorable environment allows us to begin producing those cyclins so that we can progress through G1 into S. But if DNA damage occurs, then both G1S cyclin as well as S cyclin will be inhibited. As soon as the DNA is repaired, we can then go on and enter the S phase. Re-replication is blocked by M cyclin, phosphorylation of CDC6. Likewise, the unreplicated or damaged DNA can also arrest M cyclin's activities. That would, of course, be through the Wee kinase inhibition system. And when that is released and the DNA is fully replicated and not damaged anymore, then CDC25 can reactivate M kinase and we can proceed through the G2 checkpoint. And finally, if the chromosomes line up properly and are attached to the spindle properly, then APCC can allow us to pass through the M checkpoint. Make sure that you understand these key factors of the control of cell cycle, and I wish you well on the exam. Thank you so much for listening.